On the 28th of January 2015, a lightning bolt struck deep inside Shannon National Park in the far southwest corner of Australia. The fire that followed burned for 11 straight days. 98,000 hectares of the most structurally complex forest on the continent were reduced to bare, blackened mineral soil. The Kari trees that had dominated this landscape, the third tallest flowering tree species on Earth, 60 meters high, presiding over 6 or 7 stacked layers of vegetation beneath them, were gone. The dense thickets of scrub and sedge that had packed the ground level so tightly you physically could not walk through them were gone. And running through the soil beneath all of it, invisible and silent, the real infrastructure of the forest had been cooked out of existence. That infrastructure was a web of underground fungi threaded into every root system in the forest. These fungi do not grow above ground. They fruit entirely below the surface, forming dense little nodules, truffles, and they perform a job that the towering trees above them physically cannot do alone. The soils in this part of Australia are ancient, among the most nutrient poor on the planet. Phosphorus, the element trees need to build cells and grow, is chemically locked inside the mineral structure of the earth. The trees cannot extract it. The fungi can. They latch onto a tree's root tips, extend outward in threads hundreds of times finer than the roots themselves, and chemically pry the phosphorus loose. In exchange, the trees feed them carbon. Remove one side and the other starves. The fire removed both sides simultaneously. Here is the problem no amount of machinery could solve. Those fungi fruit entirely underground. They have no way, none, to launch their spores into the air. Wind cannot carry them. Water cannot carry them. The only delivery system that works is an animal, one that digs the truffles up, eats them, walks somewhere else, and deposits the spores in its droppings. With the fungal network dead across 98,000 hectares and the only animals capable of restoring it trapped in tiny pockets of surviving swamp, the forest was locked. How do you restart a forest when the soil is dead and the only thing that can fix it cannot get from one place to another on its own? Not by planting trees, not by scattering spores from a helicopter. The answer involved dropping poison from aeroplanes and trusting a handful of marsupials that weigh less than a house cat to do the rest. The quokka is precisely the animal this forest needs to restart. Stocky, hunched forward, round-eared, blunt-nosed. Coarse gray-brown fur, built more like a football than a greyhound. Closer in size to a rabbit than the kangaroos it is distantly related to. Adults weigh between 2.5 and 5 kilograms, but inside that compact frame sits a digestive system, built like a fermentation chamber, a specialized foregut that breaks down tough fungal tissue while leaving the microscopic spores completely intact. A quokka eats a truffle in an unburnt swamp, walks across the landscape, and drops the processed spores in a warm, nutrient-rich pellet directly onto the soil that needs them. Before the fire, roughly 594 of these animals lived within the burn zone. When researchers from the Department of Biodiversity, Conservation and Attractions and WWF Australia conducted comprehensive surveys of the aftermath, they found that 77% of known subpopulations had been erased outright. The survivors and a handful of animals huddled in deep, wet gullies and waterlogged depressions where standing moisture had held the flames at bay were separated from one another by as much as 74 kilometers of open, lifeless ground, and they were not moving. The dense scrub that had hidden them from foxes and feral cats was gone, and every meter of open ground was a sight line for a predator. Tracking data from that first year showed the average quokka venturing just 12.5 meters past the edge of its unburnt refuge. 12.5 meters. The forest needed them to cross tens of kilometers. The fire had not just killed trees, it had severed the connection between the soil and the only living system capable of repairing it, and that system was pinned in place by fear. The soil could not grow trees without the fungi. The fungi could not move without the animals, and the animals would not move while the predators were watching. The intervention that broke the deadlock came from the air, but it was not seeds or spores. It was dried meat. Each bait was injected with a compound called sodium fluoroacetate, known by its industrial shorthand, 1080. and dropped from twin-engine aircraft flying precise GPS grid lines across the burn scar and its surrounding perimeter. 5 baits per square kilometer, 4 to 6 drops per year. The logic behind this sounds reckless until you understand the evolutionary trick it exploits. Southwest Australia is home to an entire group of plants, the poison peas, that naturally produce high concentrations of the exact same toxin in their leaves and stems. Every native mammal in the region evolved alongside these plants over millions of years. Their bodies learned to process the compound and flush it out. A quokka can eat a 1080 bait and walk away unharmed. A fox cannot. A feral cat cannot. They evolved on the other side of the world in landscapes where this particular chemical simply does not exist. A dose of 3 milligrams and a speck you could barely see on the tip of your finger kills a full-grown fox outright. So when the bait hits the forest floor, it functions like a perfectly porous wall. Everything native passes through. Everything invasive does not survive. The program responsible for this chemical shield is called Western Shield. It had been running across Western Australia since 1996, covering nearly 4 million hectares of public land with over 800,000 toxic baits per season. Its stated goal, in the words of the planners who built it, was simply to recover and sustain wild populations of native fauna threatened by foxes and feral cats. That was the brief. Protect the animals. Nobody wrote rebuild a forest into the budget. Within months of intensified baiting over the Northcliffe scar, fox detections on remote camera traps began to drop. And then something shifted in the tracking data. By the end of the first year, the quokkas were still cautious. The farthest any individual ventured from unburnt cover was 186 meters. But the sedges were already coming back. Thin, fragile threads of green pushing through the ash. And in the places where those first shoots appeared, cameras started picking up movement at night. Small shadows bounding short distances, then retreating. Not far, not fast, but further than 12 meters. Every time one of those animals left its swamp, foraged in the recovering zone, and dropped a pellet of spore-loaded dung onto the bare ground, it was performing a task that no planting crew, no helicopter, no government program could replicate at this scale. The forest was not being replanted, it was being re-inoculated. They were just eating. What nobody had fully grasped until a field ecologist named Carleen Bain strapped GPS collars to 29 of these animals was how far they could actually travel. The popular image of the quokka, the grinning, pudgy tourist attraction from Rottnest Island, describes an animal with a home range smaller than a few football pitches. Bain's data from the mainland forest told a completely different story. Southern forest quokkas hold home ranges averaging 71 hectares. They cover up to 10 kilometers in a single night. One female, weighing barely 2 kilograms, an animal you could carry under your jacket, was tracked moving 14 kilometers to establish a new site. These were not fragile victims sheltering in swamps because that was all they could handle. They were long-range couriers, built for distance, locked in a cage made of predators. And the cage had just been opened. Between years 2 and 5, the system tipped. The cautious 12-meter forays became routine nocturnal patrols stretching kilometers into the recovering burn zone. As sedge cover thickened, from under 13% of ground coverage to nearly 45%, the quokkas gained the structural shelter they needed to move with confidence. And as they moved, the invisible payload they carried spread with them. The spores they deposited germinated in the ash. Fungal threads reached out and fused with the roots of struggling kari and jarrah seedlings. Tiny plants that had been trying to survive in baked, nutrient-locked soil with no way to feed. With the fungal connection reestablished, those seedlings suddenly had access to phosphorus they could never have extracted alone. The effect was rapid and measurable. Root networks expanded, water uptake increased, growth rates climbed. Within 5 years, the landscape was physically transitioning from a flat, two-dimensional burn scar back toward the complex architecture of a healthy Kari forest. 3 to 7 vertical layers of vegetation rebuilding from the ground up. The quokka population tracked the vegetation almost exactly. From the critical low in the core monitoring zones, numbers climbed to an estimated 272 by 2020. 46% of the pre-fire baseline. The animals breed fast when conditions allow, a 27-day gestation period, year-round reproduction, up to 2 joeys per female per year. They did not need encouragement, they needed cover. And the cover they were helping to create was, in turn, allowing more of them to survive, which deposited more spores which grew more cover. The loop was running in the right direction. This is where the meaning of the entire operation expanded past anything its planners had written into a budget. The Western Shield Baiting Program was created in 1996 with a single stated objective: recover and sustain wild populations of native fauna. Nobody designed it as a forestry program, but by suppressing the predators, it did not merely save the quokkas, it released them. And by releasing them, it activated the only mechanism capable of reseeding 98,000 hectares of sterilized earth with the underground fungi that the entire forest depends on to exist. By year 5, individual quokkas were being tracked on migrations of up to 50 kilometers. The subpopulations that had been shattered apart, separated by 74 kilometers of barren ash, were physically reconnecting. Isolation distances collapsed from 74 kilometers down to between 5 and 30. The animals were stitching the landscape back together, carrying fungal spores in their digestive tracts and depositing them in the recovering corridors between swamps. Each corridor that grew dense enough became a bridge to the next one, which accelerated the movement of the next animal, which seeded the next corridor. The loop was compounding. And satellite imagery confirmed it from orbit. Vegetation recovery across the scar did not spread evenly. It spread outward from the exact refuges where the surviving animals had been trapped, not from where seeds had been dropped, not from where any crew had worked. The recovery map was a movement map. So how does a forest rebuild itself when the soil is dead and the fungi it depends on are trapped underground with no way to move? The answer goes back millions of years. The fungi fed the animals moved the fungi. The chain ran for millennia without interruption, until the Northcliffe fire severed it, and introduced predators kept it severed. The 1080 baits did not rebuild that chain. They removed the one thing blocking it. And in the narrow evolutionary gap between a quokka that processes fluoroacetate without harm and a fox that drops dead from a trace of it, the entire forest recovery balanced. 5 years of tracking measured the arc from a handful of animals foraging 12 meters from cover to 272 individuals migrating up to 50 kilometers across recovering ground, with 59% of historic sites reoccupied. The projection, built on continuous monitoring data, puts full recovery, approximately 600 individuals, 100% spatial continuity, restored fungal dispersal across the entire scar, somewhere around 2027 or 2028. 13 years for a forest to functionally rebuild its underground infrastructure. Not because somebody planted it, because a remnant population of marsupials, each one weighing less than a house cat, were allowed to eat, walk, and defecate in peace. And the cost of the chemical shield that made it possible runs to 37 cents per hectare per year, less than a postage stamp to protect a piece of land the size of a football pitch for 12 months. It costs more to maintain a single lane kilometer of paved highway in Western Australia, between $8,000 and $10,000 annually, than it does to chemically protect thousands of hectares of recovering forest. But the system carries a permanent condition. The 1080 baits must keep dropping. The foxes have not gone extinct. If the chemical shield fails, the predators return, the quokkas retreat to their swamps, dispersal stops, and the forest stalls. Researchers have been explicit. Recovering zones must be shielded from prescribed burns and commercial logging for at least a decade, or the corridors the animals are using to reconnect the landscape will collapse. The mechanism itself, fungi, gut, spore, tree, ran for millennia before Europeans arrived. It will run for millennia after, provided nothing blocks it. The forest did not need humans to plant trees. It needed humans to remove the thing that was stopping the trees from being planted by something far more precise: a chemical perimeter, a digestive tract, an underground network of fungi that cannot travel on its own, and a marsupial that weighs less than a house cat moving through the dark, carrying the forest's future in its gut. What other ecosystems are locked right now? Not because the biology is missing, but because something is blocking the biology from doing its job. Drop your answer in the comments.